Now we're going to begin the study of hearing and sound perception. That's another thing that primates do really well, but you probably take it for granted. You probably take it for granted you're hearing because your vision is so good. And we tend to use vision for just about everything except for one of the most important things we do, which is communicate. Communication is almost always done through hearing, through sound production and sound perception. We've even gone so far as to convert sounds into a visual form that we use to communicate called writing. But to start off, to understand what hearing is, you need to really understand what a sound is. And I'm going to give you a detailed description of what a sound is. Let's start off with a very basic idea of a sound wave. Now, a sound wave is a perturbation of the air. You can see in this graphic here a speaker, and this speaker is producing a pure tone. You can see the wave is very, very, very regular. And what it's doing is it's vibrating. If you were to go touch a speaker or look at it, you'd see it vibrating. It's vibrating because it's got a magnet in there that's responding to electric current. And as the electric current becomes higher and lower, as the voltage goes up and down, the speaker moves. And when it moves, it has a diaphragm connected to the magnet. And when it moves, it pushes air towards you. You can do this in a very controlled, high-resolution sort of method that enables you to then hear a sound. Sounds are very, very small perturbations in the air. Now, sometimes it's hard to identify with this because, you know, when you think of perturbations of the air, you think of the wind, you think of a breeze. But think of very, very small perturbations of the air, very, very small changes in air pressure, producing very, very small waves in the air medium, and you have sound. And what you have in your ear and in your head is a method for hearing these sounds, for perceiving these small changes in air pressure, and making sense out of them. As I talk here in a microphone, recording the sounds for these presentations, I'm using a computer program that takes the information from the microphone and samples it digitally to represent the sound wave as a digital waveform. And all that means is it takes samples really, really fast of the magnitude of the sound wave. And then it represents that in a data set, which is then used later to play the sound. Well, in this graphic here on the bottom left, we've got some displays from the program. And this program can also generate pure tones, and that's what you see here. The top display is showing a pure tone that's, I believe, at 400 hertz, which is a relatively low tone. Then the one at the bottom is a display of a waveform at twice 400 at 800 hertz. And you can see where the waveform, showing its characteristic troughs and hills, is more rapid. It's more dense in the waveform below. It's twice as dense. For every two hillocks in the top waveform, there's 4 in the bottom one. So it's twice as frequent, these waveforms in the bottom one, as they are in the top. And when these are played through a speaker that's also waving at that same frequency, you hear the bottom one is a much higher pitch than the one at the top. So 400 hertz is a relatively low sound. 800 hertz is a little bit higher. It's still relatively low. Most speech sounds, for example, are in the range of 2,000 hertz, which is considerably more than the 800 that you see here. Okay, now I'm going to go through complex waveforms and simple waveforms and show you what they look like in their spectral analysis. And that way we can learn a little bit more about waveforms. But basically these waveforms are small physical changes in the air that you're going to transcribe into a sound perception. So a waveform in the air is going to be turned into a sound perception by your brain. In that way you can discriminate sounds, you can communicate, you can do all kinds of things with sound. Now the waveforms are defined by intensity and frequency. Now I've described frequency reasonably well. You got 400 hertz at the top. And what that means is there's 400 wave hills every second. And the bottom one is 800, and so you got 800 little hills every second. Now you can see how small these little changes are. There's 400 within 1 second. That's the frequency. So that's how often these hills and troughs occur. Now intensity represents the size of the hill. the magnitude, the strength of the hill, how much energy is in the waveform, is the intensity. Now we turn these two things into perceptions which have different names. Intensity is referred to as loudness. So it's volume or loudness. Frequency is referred to as pitch. Now our pitch perceptions don't exactly match the frequencies. You can imagine the apparatus that we use to hear has some error in it, and it can't actually represent all frequencies with the same accuracy. So we're very accurate in perceiving frequencies in the speech range, around 2,000 hertz, and then our accuracy drops off as the frequencies get higher and also as they get lower. Other organisms on the planet can perceive higher and lower frequencies better than we can. So, for example, whales can perceive very low frequencies better than we can. Also, elephants can perceive low frequencies better than we can. Whales can actually communicate across the entire ocean using low-frequency sounds. Animals like bats and dolphins, they can hear very high frequencies better than we can. Actually, your family dog can hear high frequencies better than we can. So our frequency perception drops off around 10,000, 20,000 hertz. We can't even hear anything higher than that. And your dog can, I don't know what the dog's perception is, but it's probably about 10 or 20% higher than that. This program I have that lets me record these sounds also lets me edit them. So if I cough or sneeze or have some awkward phrase or something, I can go back and cut it out and re-record the sound. It lets me do all kinds of things to the sound that I didn't even know you could do to sound until I got this software. It lets me change the frequency of the sound so I can make my voice go up and down. I can do all kinds of transformations of it, bend the sound around in terms of volume and everything. I can basically edit the sound because it's being represented as numbers. All these samplings of the sound wave, they represent, numbers taken over a period of time. Now, the sound that I'm recording now is being recorded at CD quality sound, they refer to that, which is 44,000 samples every second. So a sample is taken every 44 thousandths of a second of my voice, and the level, the volume that's represented by that waveform is then recorded in a data file. Well, the computer program can manipulate the numbers, just like it can manipulate any other number set, and produce all sorts of special effects on the sound. For example, one of the things that I do here after I record my voice is I do what's referred to as normalize the sound. And that makes all the sound within a certain range, because I don't want the sound, if I speak too loud into the microphone when I'm recording, it will clip the sound. The sound will be too loud, and the program, it's going past its parameters. And when that happens, you just hear this sound because it's been clipped. And I don't want that to happen. I don't want the sounds to be outside of a certain range, and so I normalize the sound. And so what it does is it takes the mean value of the waveform and makes all the other values relative to that mean value. That way I can calibrate the sound so it's within a certain range. I also want to do that because I don't want some of my sound elements here to be too loud, and then you go to the next slide and it's real silent. So I got to make the sounds all within a certain range. Otherwise, they'll sound odd or be difficult to deal with when you're listening to the PowerPoint presentation. Another thing I can do is I can do a Fourier transformation of the sound. Now, Fourier transformation of a sound is when you break the sound frequencies down into their component frequencies. Okay, so every sound wave, no matter how complicated it is, can be broken down into a set of simpler waveforms. And that's what the Fourier transform does. First, though, before I show you that, I'm going to make the contrast or demonstrate the contrast between a complex sound and a simple sound. Now, the ones I showed you before of pure waveforms, that's as simple as you can get. But you've got to imagine, in order to understand human hearing perception, you have to imagine very complex waveforms. And that's what I've got depicted here. Now, the one on the top is me saying the word or the words bird song. And so what you see there on the left, the big part of what you see on the left is erd. So when I say erd, the little part you see in the beginning is the b sound. Then the next part is erd. And then the part on the right there is me saying song. Now you can tell an S sound pretty easily in a complex waveform because it looks like noise. So when you say s for an S sound, you're making auditory noise. Now, this is a complex waveform. This is the kind of waveform that humans hear and make sense out of when they listen and perceive language. If I were to play this waveform, you would hear me saying bird song. But you can see how complex it is. It looks very little like the waveforms I showed you before, where you just got pure tones. It's very complex, and it's complex because the apparatus that I use to make sound, my pharynx, my throat, my face muscles, my mouth, and everything, they're not pure forms. They're not spherical. They're not going to produce a very systematic waveform. Even if you were to listen to the sounds of musical instruments, they would be relatively complex, and that's what gives them their unique tone. So you can tell the difference between a note played on a French horn from a note played on a trumpet because of the configuration of the French horn and its complexity, and the corresponding complexity of the trumpet. Now, the things in music that produce the most consistent pure like tones are instruments like pianos. A piano is designed to produce, inside a piano is a little hammer, a little felt hammer. It hits a string and makes it oscillate at a very consistent frequency based upon the tension of the string and its shape. And it makes the air move around the string, and you perceive that wave as a note played on the piano. However, even that note would not look as pure as the ones I showed you before. Now, the bottom one is a recording of me trying to sing a note ah. So I'm trying to sing ah and make my sound very consistent. Now, you can see there that actually it's not too bad. It looks sort of pure tone-like, but obviously a human trying to make that tone is nothing like, sounding a tone in a piano. Now, you can see, however, that there are very consistent hills there, very consistent troughs with some complexity, but it's not anywhere near as complex as the top one. But again, what I'm recording is the waveform. What this program does is record the waveform. It's recording a little sample of the sound wave every 44 thousandths of a second, and recording that as data. And it's being depicted here in these graphic forms. Okay, but the point is that complex waveforms are made up of smaller waveforms, or less complex waveforms. You can take any waveform, no matter how complex it is, and break it down into pure waveforms of various frequencies that, when they're blended together, produces the complex waveform that you see recorded here. Okay, in the next slide, I'm going to show you the Fourier transforms and what they look like. Here's a spectrogram, a Fourier transformation, of the sound element at the top. Now, a spectrogram, or this Fourier spectrum analysis, it's depicting which frequencies are in this sound and what their magnitude is. So as you look across this graph, where you see the peaks represent where frequencies are in the sound, which frequencies are there, and how prominent they are. So as you look across this graph, you can see that the low frequencies, there's some low frequencies that are prominent. And they're actually quite prominent in the sound. What I'm referring to as low frequencies is, say, anything around the 400, 500, say up to a 800 range. And you can see there are peaks there. There's a peak at 500. There's a peak at, looks like 450 or so. It's hard to tell because the graph at the bottom doesn't have many increments on it. But you can see where there are frequencies in this sound, and the decibel scale on the left is telling you how prominent they are in the sound, how loud they are in the sound. And you can see this sound is actually quite complex. There's not a lot of consistency or regularity to it. You can see actually there's some regularity to the low tones. So probably bird, sounds like almost a pure tone, that I might produce. Song, ong, it sounds like something that might be lending itself to a pure set of frequencies. But this is, I'm kind of stretching it. You can kind of see in the graph at the top, or the waveform at the top, that there is some consistency in parts of my utterance there, but it's pretty complex. You can also see that there's a lot of little variations in there that represent my voice and represent my particular production of that phrasing. Things like an accent. I mean, if I was from some other part of the country, if I had learned English as a second language, I might pronounce this differently, and you would see a different waveform. Okay, but this is showing you a Fourier spectrum analysis of a complex waveform, and it's showing you all the frequencies that are in the sound. And you can see they're quite diverse. There's a lot of different frequencies in the sound, all represented at different levels. Okay, here's the spectrum analysis, or the Fourier transformation, of the second sound in which I was trying to sing ah. And you can see the waveform there at the top. And you can see a lot of regularity to it. It's a lot more than in the complex sound before, but less regularity than in a pure tone. And no matter how hard, the greatest opera singer tries to sing a single note, it's going to have complexity to it because that person trying to produce that note is not a musical instrument. There's a lot of complexity to our apparatus for making sound, and a lot of error, a lot of noise, a lot of other tones get in there, and I think it actually lends itself to the production of very naturalistic sounds. Singing would not really be singing if it was produced perfectly, as if you had an apparatus like a robot might to produce pure tones. Now, if you look at the Fourier transformation of this sound, you can see the frequencies represented are much more regular, especially in the lower part, lower than 1,000 kilohertz. You can see where we're beginning to get a very systematic set of frequencies. Now, what this represents is, if you look at the graph on the top there, you can see there's a major frequency. You can see it there. It's the big hill. Then next to it are some smaller ones. And then within the big one, there's some dips. There's some parts to it. It represents other frequencies in that sound. Now, the regularity that you see here, so you see intervals, it looks like it's starting at about, I guess, say that's 100, let's say it's 200 hertz there, the first peak. Then the next peak is around, looks like 400. Then the next peak is around maybe 600. And then there's 800 and so on. So it looks like every 200 hertz there's a peak. Now, this is representing a primary note, a primary tone here, and I don't know what it is. I'm not, I don't know enough music to know what a 200 hertz tone translates to. I do know that a 440 hertz tone translates into the note A. A on the piano or A in music. That's 440 hertz. Well, this is showing me that I have a bass note here of about, I'm going to say 200 again. It may be 300 or something. It's hard to, since the graph at the bottom doesn't have many marks on it, it's hard to say what where the peaks exactly are. But the point of this graph is that you can see where there's a periodicity to the peaks. And what I have here is a root note, say it's at 300, and then every 200 after that I see a peak. Well, these 200 hertz increments after that are representing overtones, what's referred to as an overtone to the bass note. So when you see peaks at regular intervals from a bass note, that's referred to as overtones. Now, that's very common in singing, for example. It's common in musical instruments. If you were to take a piano, and pianos have a stop that keeps the strings from responding until you pluck them. But if you release that with a pedal, they have a pedal at the bass that releases that, and you play, say, a note C on the piano, that will cause all the other C notes on the piano, all the other C strings on the piano board there, to resonate with that C. And they represent the overtones of that original C that you pressed the key on, you started from. So the C's, all the C's want to respond, but also other frequencies will respond. They're also overtones to the C. And what you've got are resonant frequencies in which the airwaves are making the strings, when the sound hits the string, that string begins to resonate with that frequency and make its own tone. So some of the energy from the sound wave is transmitted to the string. It starts to vibrate and you get overtones. Well, what's happened here is I produced overtones. So the sound that I'm actually making is a composite of regular frequencies that, I've interpreted as whatever tone I was trying to make. but the tone I was trying to make is still relatively complex. It's not a pure tone. It represents a bass note with overtones that are pure-like. They're more pure than a complex tone, like when I said the words bird song. But it's still quite complex. And this spectrum analysis is telling me that there's some regularity to this, and there's a lot of complexity. Now, if you look at the right part of the spectrum analysis, you see there's a lot of frequencies being represented in the sound that are probably unique to my voice and things like that. Now, it's important to understand the basics of a spectrum analysis because that is what your hearing apparatus, with the basilar membrane, the cochlea, what your ears do, is they don't actually send a sound wave to your brain. Your ear apparatus will do a spectrum analysis of the sound, and that information is then sent to your brain. So your brain does not get a direct representation of the sound wave. It gets a spectrum analysis of the sound wave done with a biomechanical device that's referred to as the basilar membrane or the cochlea, in a very remarkable invention of evolution in order for this to happen. But that's why it's important to understand that sound waves can be broken down this way, can be analyzed this way, because this is what your hearing apparatus is going to do as its first step in perception. Well, the first step to understanding hearing is to figure out how your ears work. And here is a graphic depiction of the outer ear. Now, the outer ear is characterized by the peni, or the pinna. These are what you refer to usually as your ears. The peni, they end up reflecting the sound wave into the auditory canal. It's important to keep in mind that the peni have their own unique shape. As I'm sure you're aware, everybody's ears look a little different. What happens then is that the sound wave is reflected into the auditory canal slightly differently by each person. And later we'll talk about how that influences your ability to localize sound, but it has a big impact on your sound perception. The sound wave is now still in the air, and the sound wave is conducted by the peni into the auditory canal. The auditory canal is this tube that going to go into the receptor apparatus, and it represents the first stage in processing the sound. So the peni and auditory canal represent the first sort of filtering of sound. The sound wave does change, of course, when it bounces off the peni and reflects into the auditory canal. It is a reasonable facsimile of the sound wave that's in the air, but obviously these structures will change the sound slightly. And then the sound wave is conveyed to the eardrum, and the eardrum is where the first representation of the sound is made by your anatomy. Okay, the next stage is for the sound wave to impact the tympanic membrane, your eardrum, and I think you're all familiar with them. It's a tissue. It's a skin tissue that's stretched across the opening there. It vibrates with the sound wave, and so it's got to be quite fine. It's got to be quite thin and fine there in order to respond to the sound wave. Behind it is an opening. The middle ear is an area that's also filled with air. And this air is conveyed by the eustachian tube from your throat. And so you may recall when you clear your ears if you're in an airplane, or if you do any kind of diving, scuba diving or something like that, you have to clear your ears. And what you're doing is equalizing the pressure across your tympanic membrane. Obviously, if you don't do that, you're going to cause the tympanic membrane to be injured. It might even rupture. And that's what the eustachian tube is for, is to convey air pressure from the ambient air. It's actually in your mouth, but to equalize the pressure across the tympanic membrane. Now, connected to the tympanic membrane are three of those famous little bones. A big trivia question is always, what are the smallest bones in your body? And I guess it's the stapes is the smallest one. But these three little bones convey the sound wave to the cochlea. Now, what you've got to do is basically convey a sound wave in the air has got to be translated into a wave in fluid. Now, this is common actually in all your sensory systems. There's this translation of the external world for us, which is filled with air, into the fluid-filled world, which is our bodies. As you might think, you're basically a big sack of colloidal fluids. You're a big sack of fluids. You're constantly communicating with the air environment in various ways. But your neurons the nervous system cannot live in air. It's got to be in the fluid all the time. So anything you're going to perceive, be it light information or sound waves, touch, all kinds of things like that, have to be conveyed from an air environment into a fluid-filled environment that your neurons can live in. There's no better example of that, I don't think, than hearing. See, if the neurons could live in air, they would just be connected to the tympanic membrane, or there would be a membrane there quite similar to the basilar membrane that was just in the air. And the air sound wave, would communicate with that membrane, and you wouldn't have to have these bones and this whole apparatus here. This apparatus is designed to amplify the sound wave so that it's better perceived later on. It's got to be a little bit bigger for the neurons to respond. And it's also there to convey a waveform that's in air into a waveform that's in fluid. And I'll get to that more in detail in a second. The amplification is about 20 times, so it actually amplifies the air sound wave about 20 times in order for it to be transmitted and communicated to the fluid. Okay, well, the next step is the inner ear, and we'll talk in the middle and inner ear, and then we'll sort of complete the cycle or complete this diagram of anatomy. One of the things that happens in your middle ear is there's a kind of shock absorber system for loud sounds. And you can see another depiction here of the middle ear, where you've got the three bones, and you can see that there are small muscles connected to these bones and to the ear apparatus that stops them from moving too far. So if the progress of the bones is too great, indicating the sound is too loud, then the muscles will contract and pull things back together and not allow the bones to move too far. If you hear sudden loud sounds, and a lot of times these muscles actually protect us from our own voices, because you can imagine if you're shouting, you're producing some of the loudest sounds that your system's going to hear because it's so close to the system itself that you need some kind of shock absorber to keep the system intact so it doesn't respond too far. And this is referred to as the attenuation reflex because it attenuates the movement of the middle ear bones. Okay, here's our first view of the cochlea. The cochlea is the structure that's going to be perceiving the sound and conveying the information up to the brain. It's the sound receptor organ. And it consists of fluid-filled space. There's really two components to it. There are two fluid-filled areas. And then in between them is a membrane. This membrane is going to respond to the sound wave in that fluid-filled medium. But right now, just take a look at the cochlea to see how it's structured. You can see it's curved like that to save space, to be efficient in taking up space there in your ear. But you could stretch it out and it will still perform the same function. It's just that it's like this in order to conserve space. Basically, it represents the interface between the sound wave in the air and the auditory nerve, which is going to convey information about sound up to your brain. Here's a cross-section of the cochlea showing the three chambers. There's two major chambers, and there's a third one. The third one makes up the organ of Corti. So what you have are these two fluid-filled areas, and then in the middle you've got the basilar membrane, the tectorial membrane, and all that's referred to as the organ of Corti. So you've got three areas, the three chambers, two major ones, one minor one, and then you've got the organ of Corti in the middle there. Now the interaction between the sound wave and this being propagated in this fluid with the basilar membrane is going to produce perception. But here's a cross-section of how it's basically structured. Okay, let's go into even more detail about this organ. You can see the two chambers, the two major ones, the top and bottom, and the one in the middle. Notice that you've got really two membranes here, two areas you need to be concerned about. The bottom one is the basilar membrane that's not colored there. The top one is the tectorial membrane, which is colored in yellow. In the basilar membrane, you have the hair cells that are going to be the sound receptors. They're going to be the receptor that's going to respond to the sound wave. The tectorial membrane lays on top of them and helps them respond, The whole apparatus there is going to vibrate with the sound wave and produce activation, presumably of the auditory nerve in that spot. But that's the relationship of the hair cells to the tectorial membrane, and I'll go into that in more detail in the next slide. Okay, this is the first slide in depicting how the basilar membrane responds to the sound. There you see it up at the top there. Now those sort of curly areas at the top actually represent your vestibular system, your balance system. The cochlea is just the curled up part at the bottom. They both systems share the same fluid-filled space because they both use fluids to perceive. Right now, though, we're just going to go through the cochlea. We'll eventually get to the balance system a little bit later in the course. Okay, well, there you see in the bottom, if you had stretched out the cochlea, the basic layout of the relationship of the basilar membrane in this very schematic form to the stapes and to the chambers that convey the sound wave from air to fluid. So as the stapes is moving there with the sound wave, now remember the sound wave hits the tympanic membrane, makes those bones move, and then the stapes is going to move against the oval window. And that movement against the oval window is going to cause the fluid inside the cochlea to respond consistent with the sound wave. Well, the basilar membrane is sitting there, and it's going to respond in a characteristic way to that wave that's propagated in the fluid. Well, here's the basilar membrane stretched out again. So you've got it open now, stretched out completely, just for schematic form. Obviously, it's all curled up still in your actual ear. There's the stapes up there, pressed against the oval window. The stapes is vibrating with the sound wave. That wave is then communicated through the endolymph all the way through across the basilar membrane, all the way around it, and then over to the round window. The round window absorbs the energy that might still be in the fluid wave. It gives the system a way of responding so it doesn't get overwhelmed by the size of the wave. Now what's going to happen, though, is the basilar membrane is going to respond to the sound wave by its place location along the basilar membrane. But as you move, the basilar membrane is going to fluctuate with the sound wave. It's going to move up and down with the sound wave, and the neurons that are connected to it are then going to respond. But basically what the basilar membrane does is it moves up and down with the sound wave, and that causes things to happen that result in perception. Well, here's another schematic of the basilar membrane. And this is going to depict how the basilar membrane responds in different locations of the membrane. Okay, so there's the basic layout where you've got the two cavities, one above the other, the basilar membrane in between. Now notice that the basilar membrane is not continuous all along its length. It's sort of, it's narrow toward the stapes end, broadens out toward the far end, but also it's I believe thinner. Yes, it's thinner at the near part and thicker at the far part. Now what that means is that the membrane is going to respond differently to different frequencies of sound. If a sound wave or a wave goes through that medium, the fluid medium, different parts of this membrane will vibrate in different places depending upon the frequency. So here we've got a frequency distribution over here on the right showing you what frequencies and how they respond. Now when you send a low frequency through this medium, the area at the far end of the basilar membrane will vibrate the most. The area of the membrane closest to the stapes will not vibrate. Well, it will vibrate, that area will vibrate, however, when the sound wave is at a high frequency, and the far end will not vibrate. Now this is going to be very useful in conveying information about a sound wave to your brain. Because where this place is, where the location of the vibration is going to stimulate certain neurons and not others. Your brain then gets information about where on the basilar membrane it's vibrating, and it ends up getting a spectral image, a spectral depiction of the sound. And you can see where that's beginning to happen. Now, so we've got a membrane that's structured in a way that it will respond differently to different frequencies by different places along the membrane. Now, this is a remarkable invention of evolution. For this to evolve like this, so that you actually have a apparatus, a biomechanical apparatus that produces this phenomenon. It's just amazing. For it to be invented by evolution, for it to work so well. Now, obviously, it gets damaged a lot in disease and things like that. It doesn't work so well, but it's amazing that it works so well for so long and is resilient as it is. Okay, well then I'm going to go into more detail about how the basal membrane responds. But this graphic is important to convey that different parts of the membrane are going to respond differently to different frequencies of sound. Now we go back to the organ of Corti in the basilar membrane. Okay, so the basilar membrane is in the bottom of this. It's the base for this organ. And the basilar membrane now is going to be responding differently at different parts of the basilar membrane depending upon what sound frequencies are there. Well, here's the receptor system that responds to that movement. So it's going to convert that movement into nerve impulses eventually through the interaction of the tectorial membrane and these hair cells. Okay, so here's the basic anatomy of it. You have outer hair cells, which are sitting out there more in the central part of the basilar membrane, and they're going to be responding with more energy to the wave itself when it passes along the membrane. Then you have inner hair cells, which are going to respond differently to the sound wave. Basically, the outer hair cells respond a lot, the inner hair cells respond a little. And they interact with each other, and I'll get to more detail in a second. But basically, here's the layout. You've got the organ of Corti. It consists of the basilar membrane, the inner and outer hair cells, and the tectorial membrane. And we'll go into more detail about what happens in the next slide. Okay, here's a photomicrograph of the hair cells. You can see their configuration here. So there's the basilar membrane sitting there, the organ of Corti. Along the top of it are these structures of hair cells, the outer hair cells. Now you can see that they're in clusters. They're not continuous. This turns out to be important into how they respond. Also, the tectorial membrane here has been removed. So you see these clusters of hair cells. These cilia are going to respond by physical motion, and that physical motion is going to be translated into a neural impulse. This is basically what they look like in a little more of a three-dimensional depiction, where you see them in clusters like this, because they got to respond in a certain way to make the gating happen that lets ions in and out of the receptor and enables them to respond. So they can't be continuous. They got to be in these little clusters, these little sort of, these little sort of pyramid shapes here, or these little curved shapes. Well, here's what basically happens. Here's another depiction of the schematic here, the organ of Corti in the basilar membrane. And you can see the inner and outer hair cells depicted in graphic form here. Well, the basilar membrane now is going to move up and down with the waveform that's coming through there from the sound wave. When it does this, the cilia, especially the outer hair cells, will push up against the tectorial membrane. So that membrane is laying right above them. When the basilar membrane moves up, the tectorial membrane moves against the hair cells, the cilia, and makes them move, makes them physically change. And depending upon the power that's in the waveform at that point, at that spot, you'll see a lot of movement of the hair cells or less so. But this is crucial for the hair cells to respond. And you can see where the tectorial membrane is playing a role here of essentially amplifying or filtering the response so that you get movement. If the tectorial membrane was not there, the hair cells wouldn't matter. They wouldn't have anything to move against. They're not going to be the whole hair cell itself will move, but the cilia will not move relative to the hair cell. And this turns out to be crucial for the hair cell's response. Okay, here's the basic shape of a hair cell, of an inner and outer hair cell. Now, what we should pay attention to mostly here is the cilia. The cilia are these small hairs that come out of the cell. They're going to be moving against the tectorial membrane when the basilar membrane goes up and down. But here you see the cell's basic structure. You see a nucleus of the cell, the membrane, the cilia coming out of the cell. Now notice that the inner and outer hair cells are slightly different. The outer hair cells are more perpendicular to the surface of the basilar membrane. The inner hair cells are a little more shaped. The most important difference, I think, between them is the fact that the neurons that connect to these cells are much more dense for the inner hair cells than they are for the outer hair cells. That turns out to be important in calibrating the response of the basilar membrane and in interpreting the waveform that's coming through that fluid. Well, let's take a look at how this works, how this depolarization of the cell occurs using these cilia. Now, this is a schematic here also of the cilia, showing 3 of them, the very tops of the hair cells. And in these cilia, you have protein chains that are embedded in the membranes that function as gates for ion receptors. So you have channels for these ions. And when a hair cell moves, you have this gating of the a channel. It's mechanically gated. Now, most of the gates for ion channels in the nervous system are done by a bioelectrical processes, essentially. They're not usually mechanical like this. They're mechanical in this system because the fluid is going to be physically moving. It's going to be physically moving these hair cells, and it's going to be changing the potassium channels because of mechanical forces and not electrical magnetic forces. So what happens is when a cilia move, this only responds, of course, to one direction. And when the cilia move in a direction that opens the channels, potassium flows into the cilia. So that's all we have to keep in mind now. Something's going to happen after that occurs, but right now we just need to know that when the cilia move in a certain direction, the potassium channels will open, potassium will flow in. When they move in the other direction, the potassium channels are closed, and potassium will not flow in, causing a response of the receptor. Okay, but it's important to keep in mind we've got a system like this where we've got mechanical channels that are opening, closing, and letting potassium in or not. Okay, here's the next phase of the operation. Here's the hair cell in schematic form. So you see a very basic image of 3 cilia up there. The cilia have been pushed to the correct direction, allowing the potassium channels to open. So the basilar membrane has come up, they pushed up against the tectorial membrane, and they pushed against it in the direction that allows potassium channels to open. Okay, potassium flows into the cell. It flows into the receptor cell. Now, when it flows in, it doesn't just cause a depolarization of the postsynaptic neuron. What it causes is a depolarization of calcium channels. Okay, so calcium channels are going to be gated here. Calcium flows into the cell, and this causes a neurotransmitter to be secreted across the gap from the receptor to the ganglion cell, neuron. So, and that depolarizes that ganglion cell. It then is depolarized, and information is conveyed up the system into your brain from that neuron. But there's the basic process. You have mechanical forces that cause these mechanical gates to open at the top of these cilia. The potassium flows into the cell. That causes depolarization of a voltage-gated calcium channel. Calcium flows into the cell, and calcium causes the secretion of the neurotransmitter. The neurotransmitter is probably glutamate in this system. But at any rate, it's sort of this 2-step process where you have potassium gates open at the top, potassium flows in, causing depolarization of calcium channels, allowing calcium to flow in, and then you have neurotransmitter release prompted by the calcium. So this is how this mechanical force of the waveform in the fluid gets translated into a response in the nervous system. Okay, this is a graphic that depicts the relationship between the outer hair cells, the inner hair cells, and the neurons that are connected to them. Now, there's an obvious difference in how the inner and outer hair cells are configured in the nervous system. You can see where the convergence, see the 3 cells, the 3 outer hair cells, have their neurite connected to just one neuron. So 3 of them are going to be out there responding and clicking away, but only causing a neural response in one neuron. Now the inner hair cells, if they get stimulated and they depolarize, then you've got probably hundreds of neurons connected to that inner hair cell that are going to respond. So there are many more hair cells that are going to feel the effects of an inner hair cell moving than feel the effects of an outer hair cell moving. This relationship between the inner and outer hair cells and how they're responding is very important information for the nervous system. It probably is related to the conveying information about sound volume and things like that. It's still quite a mystery how this works. The relationship between inner and outer hair cells and their firing patterns and how they how these are interpreted by the central nervous system is still not known. But clearly there's a big difference, and the inner and outer hair cells working together produce the sound perception. One of the things that the receptors do when they're stimulated is they will sort of jump up and down with little motor proteins that are embedded in their membranes that allow them to actually exaggerate their response. So when they're stimulated, they will actually move with the sound, and this will cause the potassium channels in their cilia to be more depolarized, to be more open, and allowing more depolarization of the calcium channels inside. So you got to imagine that the response when the cilia are stimulated gets amplified by this process of pushing against the tectorial membrane, causing the response of the receptors to be greater than it would be if it was just relying on the sound wave itself in the basal membrane alone to push up against the cilia. So what happens here, you have kind of a trigger, kind of a, there must be some sort of threshold that makes this work. And it reinforces or enhances the contrast between where the basal membrane is moving with a lot of energy versus where it's not moving at all. And so your nervous system response gets filtered by that. So there's a contrast now between the frequency, the part of the basal membrane conveying a certain frequency versus the others. And so you have a rarefication of the sound, not unlike the kind of contrasting that's done by the retina for certain light frequencies. It's a completely different process, but the end result is similar in the sense that certain neurons are stimulated in a more than they would be otherwise if you were just relying on the other process that's has much less energy in it, doesn't make the basal membrane move as much. Here you've got, once these hair cells are stimulated, they sort of fire, they sort of move with the sound, causing the depolarization of neurons that they're connected to, more so than if they were just using the mechanical forces alone that were there. Okay, let's get back to the basal membrane again. The basal membrane, as you can see it here, another schematic. It's got this odd sort of shape to it. And what's going to happen is the sound wave is going to be propagated by the stapes into the fluid of the cochlea. That sound wave is going to travel through this fluid. Now, the basal membrane is designed in a way that high frequencies make it move more toward the stapes. Low frequencies will make the other end move more. Now, when these movements occur, they move in what's referred to as an envelope of movement, which has a curve, almost a bell curve to it, where you've got a lot of movement at the characteristic frequency or the middle part of the frequency range, then less movement down the sides. Now imagine this happening very, very fast, though. Imagine that with the complex waveform, how many places along the basal membrane that might be moving, because there's frequencies of that sound, frequencies associated with that location in the sound. But anyway, it moves within an envelope of movement, where you have a lot of movement where the characteristic frequency is, and then less movement in frequency ranges that are above and below that. Here's a graphic depiction of the characteristic frequency ranges for the basal membrane, showing its degree of movement in different places associated with different sound frequencies. So 25 hertz, which would be a very low frequency, makes the very end of the basal membrane move, but then there's no envelope on the other side. So that's the lowest frequency that you can perceive. Then as the frequencies move up, you can see where now there's a above and below envelope around the characteristic frequency for that part of the membrane. So say at 200 hertz, you can see where there's a a peak there of movement at 200 hertz. It rapidly drops off at the far end. Then there's a very gradual dropping off of the envelope toward the higher frequency. Now, as we get up into the frequency ranges that we perceive well, like the 1600 hertz, see 1600 hertz is almost within the range of the speech range. You can see where there's a very characteristic, up and down form to the movement of the basilar membrane. Now, this contributes to the place model for frequency distribution in the basilar membrane. Now, remember that at each spot along here in the basilar membrane, there's the organ of Corti with a tectorial membrane laying over the cilia of the hair cells. Now, you can see if that's the case, a low frequency, a very low one, say even at 200 hertz, is going to be stimulating a lot of hair cells along that range near the apex of the basilar membrane. But a higher frequency is only going to be, say at 1600 hertz, is only going to be stimulating a relatively small number of hair cells. That enables you to perceive sound frequencies in that range with much more clarity and with much more higher resolution. You can tell the difference between a 1600 hertz sound and a 2000 hertz sound very well, because you have this very marked peak at the two different places on the basilar membrane where the neurons are firing, indicating that those frequencies are in the sound or not. Well, this leaves us with a very compelling model of how the basilar membrane works, how the organ of Corti works, how the cochlea works. What happens is you have parts of the basilar membrane that respond very strongly to single frequencies. So here you see a graphic depiction of this, where you have a characteristic frequency for a certain part of the basilar membrane. It looks like 1800 hertz. So at that frequency, you see a very peaked response of the basilar membrane. When that curve comes through at that frequency range, it makes the basilar membrane move in that spot, and that spot only. Well, this gives rise to the place theory, or the topographical model of how the basilar membrane works. Here you see it curled up there, and what you see are the spots on the membrane that respond to different frequencies. So you can see early on you've got responses to 7,000 hertz, 6,000, 5,000, 4,000, and so on. You can see as it moves toward the apex, or the end of the basilar membrane, the frequencies start out high and go to low. Well, when neurons are responding in each one of those places, your central nervous system is getting specific information about which frequencies are in the sound. And this is the this is the mechanism that gives the brain a spectrogram of the sound. Essentially a kind of Fourier transformation of the sound into component frequencies. Because suppose the sound has 7,000 hertz in it, 2,000 hertz, 600, 700, and 800 hertz. Well, those areas of the basilar membrane will all be active while that sound is present. The neurons in that spot will then be responding. So what your brain is getting is patterns of responding consistent with where the neurons were in the basilar membrane. So what your brain gets is a spectrogram analysis, a spectral analysis of the sound. That's what gets mapped onto the auditory cortex up in your brain. So the sound wave itself is not represented in the brain. A spectral analysis is what's sent to the brain about the sound wave. The same kind of Fourier transformation that I did with a mathematical process, your hearing apparatus does, the cochlea does, and the basilar membrane does with this place model, and the unique structure of the basilar membrane, that it only responds in different places to different frequencies. Okay, here's another depiction of this. Here on the left, you've got a complex waveform, a complex wave tone. It consists of 440 hertz, which would be the note A, 880 hertz, which would be the note A in a higher octave, and then 1320 hertz. And that's what the form would look like if you blended those three pure forms, those pure tones, into one wave, blended them all together in the sound. That's what it would look like. So if you had a program like I'm using to record my voice, and you played those sounds three together, and see, this program can actually be used to blend these together. You could create three waveforms of those shapes, of that periodicity. You use a blending tool, and the computer will blend the two forms together, and presumably it would look just like that. Okay, well anyway, that waveform hits your outer ear, hits the tympanic membrane, causing it to vibrate consistent with the energy that's in this waveform. The bones in your middle ear respond. They transmit the wave into the fluid of the cochlea. The basomembrane is then going to respond in different places based upon the frequencies that are in the sound. And there you see it as in a kind of schematic there. So you get the high frequency end, the low frequency end, and the basomembrane. And there you see where 440 hertz, that's lower, is in red. And then 880 is in green, and 1320 is in purple. Now what that means is that those places on the basomembrane are responding to the sound, and the other places in the basomembrane are not responding. So all the neurons in the purple, the green, and the red areas there on the basomembrane are firing away there. The cilia are moving, they're depolarizing, potassium flowing into the receptor, calcium flows into the cell, you have release of glutamate, the neurons are responding, and what your brain is getting then is a pattern because the others aren't responding. So the other neurons connected to other parts of the basomembrane aren't moving, aren't responding, and so your brain is getting a spectrogram of the sound, of what frequencies are in the sound. And of course this gets very complex. Sounds can have lots of frequencies in them, as you saw with the analysis I did with my voice samples. So you can imagine, and of course over time this changes very rapidly. So our perception of sound is being done, this is really being done on the fly really fast, but it's being done by different patterns of spectrograms being sent to the brain. Your brain then formulates and constructs a perception from those patterns. This is a graph showing you that the neurons connected to the hair cells are responding to characteristic frequencies. So when a neuron is connected to a hair cell, when the hair cell is displaced by the movement of the basomembrane, there's a depolarization, a neurotransmitter goes across the gap, and the neuron becomes active. And here you see an electrode attached to the neuron. And the graphic is showing you its characteristic frequency, which is 1500, and how it responds when that 1500 hertz sound is played into your ear and played into the ear. Now, it's also showing you that volume is being encoded by the firing rate. So the number of spikes is greater with the higher intensity of the sound at that frequency than with the lower intensity. Now, this is the dominant model of how volume is conveyed to the nervous system by firing rates. There's problems with this model, but certainly this occurs when a sound is louder. You have more firing of the neuron than when the sound is softer at its characteristic frequency. So the nervous system gets not only a spectrogram of which frequencies are in the sound, but by the firing rate of the neurons at those places in the basomembrane, it gets information about how strong the sound is, how loud it is. And presumably as the basomembrane displaces the cilia even more, there's more depolarization, and so the firing rate increases to convey that the sound wave at that spot is large, indicating that it's loud. However, loudness is something your brain constructs. So it's not something that's actually in the sound wave directly. Again, this is a kind of sampling of the sound wave, like the computer is doing with my voice now. It's conveying information about the sound wave, which then is reconstructed as loudness in the brain. And this was dramatically represented by a case that I did quite a while ago of a patient that had a misperception of loudness from, basically the patient had a stroke in the right hemisphere that caused him to neglect sound in his left ear. Because as I'll get to later, the sound is conveyed across your brain. So your left ear goes into your right hemisphere, your right ear goes into your left hemisphere. And this patient had a stroke in his right hemisphere. And what happens is the intact side will dominate the damaged side. And in his case, if we played sounds that were in both ears, he would only behave as if he heard sounds in his right ear, and the sounds in his left ear would get suppressed. However, if he played sounds just in his left ear, he would hear them, and he would hear them fine. But But when you played sounds in both ears, the right ear that was intact, that was healthy, would suppress the unhealthy, or the side that had been damaged. Well, we were very interested in what happens when you manipulate loudness. Suppose you turn the volume up really high in the left ear, the one that he's neglecting. Well, will that compensate for the suppression that's occurring by the intact side? Well, we turned the volume up as high as the equipment would go, and he still behaved as if he was only hearing information in his right ear. However, as soon as you turned off the right ear, he heard the sound in his left ear as if it was extremely loud, which it was. He heard it like wham, it's really, really loud. But until that point, until you turned off his right ear, he behaved as if he'd heard nothing in that ear. Well, that indicates that the brain is operating on this as information. There is no sort of power function that you can equalize by playing things louder in one ear than the other. What's being conveyed to the nervous system is a firing rate, a pattern. It's information, data is being conveyed to the central nervous system, and not a facsimile of the sound wave that has a power or energy in and of itself. Just because a neuron's firing more often doesn't mean that it's somehow conveying more energy into the central nervous system that can balance out or compensate for losses in other ways. But at any rate, the information that's conveyed about volume is probably conveyed, a lot of it in firing rate, but still there's some mysteries to be figured out about how volume is encoded in the central nervous system and how you calibrate it in your actual perception. So in this graphic, you see a pattern of frequencies along the bottom there, and the characteristic frequencies of different auditory nerves. Now, what this graph is conveying is that at higher frequencies, at about looks like it's starting around 2200 hertz here for the first graph, you see a very strong peak for the higher frequencies. They're very well discriminated from each other. But as you go below 1000 hertz, you can see where you could discriminate, 1000 from 800, but you can't discriminate 1000 from 950 or something. The neurons just aren't going to respond with enough resolution for you to discriminate sounds at that difference of frequency. And this corresponds to the psychophysical studies of frequency. The lower frequencies are harder to discriminate from each other than higher frequencies are. Now, obviously, there's a point in the real high frequencies where you also can't discriminate. There's a certain point at which you can't even hear high frequencies. And so there's an optimum range of frequencies that we can discriminate well. And these turned out to be the frequencies that we use for speech. These are the communication frequencies, the frequencies that most other organisms communicate or make noises in. This is the range that the natural environment mostly presents us. And obviously it was relevant for primate evolution. So it's a range of approximately, it looks like 2,000, all the way up to maybe 20,000, maybe 30,000 hertz. We can make fine discriminations. But when it gets above and below that, our discriminating ability goes down, and it also corresponds to the firing of neurons. The basal membrane just cannot respond, across every frequency, and so it sort of breaks down at a certain point, and our discrimination gets less and less. Now, one of the problems that we have in the science of studying of hearing is that the system, even though it's quite good, isn't matching the discrimination of frequencies we can actually make. So if you mapped out all the characteristic frequencies of different neurons in different parts of the basal membrane, you come up with quite a range of frequencies we can discriminate. But it turns out that we can discriminate many more. And so the problem was, well, if this system, the place model here, doesn't account for all the discriminations we can make of frequency, how else do we do it? Well, one prominent theory has to do with the phase locking of neurons to different frequency distributions or different frequency models. And here you have a depiction of that in which the nerve firing bursts. You get neurons that are firing in bursts consistent with the shape of the waveform. And so here you've got neurons firing. We play a 500 hertz pure tone. That's a relatively low frequency. You can see a firing pattern associated with that, and the neurons they don't burst much with a 500 hertz tone. Now you can probably discriminate a 500 hertz tone from say a 600 hertz just by the basal membrane's response alone. It doesn't need to be phase locked, but as you go higher and higher in frequency, and you want to make finer and finer discriminations, you have nerve firing bursts that are associated with the shape of the waveform. So here you have a 1,000 hertz tone. So these time intervals that are depicting in this graphic are 1/1000th of a second. Well, 1,000 hertz tone is 1,000 of those hills for every second. Okay, so every 1/1000th of a second there's a peak. And what ends up happening is the neurons will begin firing according to these peaks. So every peak that occurs, it'll start firing. It'll start bursting. And now if you could imagine it bursting at different hertz patterns, you can actually end up discriminating much better between different higher frequency pitches, higher frequency pure tones anyway, from other higher frequencies. And so you can discriminate better than if you're just using the place model alone. So the bursting pattern being associated with the shape of the waveform is giving more information to the central nervous system about which frequencies are in the sound. All right, well, the response to the basilar membrane clearly has a tonotopic structure to it, in that the frequencies are arranged by their place on the basilar membrane. Well, that place model gets replicated in the auditory nerve itself. So the auditory nerve that's coming off the basilar membrane is going to be laid out according to the frequencies of the sound. And then that layout is going to be replicated up in the nervous system in the cortex of the brain. So the spiral ganglion that's going to be conveying information about different frequencies. So if it's active, it's indicating, say, a 1 kilohertz tone versus 4, is then going to be mapped in the auditory nerve and all the way up the system, so it matches the frequency range that's in the basilar membrane. Now, that would mean, say, if you damage the top part of the nerve, you'd miss certain frequencies. If you damage the lower part, you miss other frequencies. So there's a tonotopic map to the auditory nerve, and that gets conveyed up into the nervous system. The cortex of the brain is also mapped that way and corresponds exactly to the frequencies that are depicted in the basilar membrane. And we will see this again in other sensory systems, where frequency data in particular is mapped. Just about all the sensory systems use mapping to convey information. So where you are in the system, which neurons you're referring to in the system, will give you information about what features of the stimulus are being perceived. So you see mapping, obviously in the visual system there's mapping. And then in the auditory system, frequency is mapped. And then later we'll learn that your touch and those sensations are obviously mapped by the shape of your body. The taste and smell are even mapped. So properties of the sensory systems and how they perceive things are mapped. And that's something that the nervous system uses to record and perceive information. Okay, here's a summary of how the auditory system basically works and how you process sound frequency in the cochlea. Okay, so a sound pressure wave is communicated to the cochlea through the middle ear and the three bones in the middle ear. They hit the round window, they started vibrating. The sound wave is then propagated in the fluid. The basilar membrane starts to move consistent with the sound wave. And the place in which it's moving is consistent with the frequencies that are in the sound. The outer hair cells, cilia, they bend with a sound pressure wave in the basilar membrane, and they cause the system to begin to respond. And there's a modal response of the outer hair cells that amplifies the basilar membrane's response. Remember, they move back and forth. These factors are communicated to the inner hair cells, and these inner hair cells begin to respond like the outer hair cells, except there's some sort of an interaction between the two, especially when the sound, if a frequency is very well represented in a sound, you'll have the inner hair cells and the outer hair cells responding. But the inner hair cells, the outer hair cells respond. They cause neurons to discharge, and these neurons respond by phase locking firing patterns that are locked to the frequencies that are being conveyed by the basal membrane's movement, and of course the frequency that's actually in the sound wave is being propagated in the fluid around the basilar membrane. So this is basically the process. It's this sort of a sequence here that looks pretty complicated. But basically what you have is a membrane, the basilar membrane in the cochlea, that's responding by place location. And the cilia of the inner and outer hair cells are responding because that location is moving a lot. Then the neurons connected to those cilia begin responding, and they phase lock to the frequency that's being conveyed by that area moving to present the central nervous system with very specific information about what frequencies are in the sound. And the neuron firing pattern and which neurons are firing. Which neurons are firing is associated with their location in the basilar membrane, so that's going to convey frequency. Then the amount of firing is going to convey volume. It's going to convey the strength that's in the waveform, causing the cilia to depolarize the neuron. All right, well now we've got a basic model for how the basilar membrane works and how this frequency information is going to be conveyed to the nervous system. Well, what happens after that? What happens, when the information is being encoded by these neurons? Where does it go? How is it processed by the brain? Well, a lot is known about how the basilar membrane works and how these frequencies are encoded, and far less is known about how the central nervous system really processes sound. Well, the first step, of course, is to get it into the brain, get it conveyed. And here you see a schematic showing the neural pathways for the auditory system. You can see the basilar membrane there, the organ of Corti, and you can see the auditory nerve coming away from the basilar membrane. There are nuclei that these neurons will synapse with. In particular, the cochlear nucleus is something that's in, one of the first places it synapses. Now, the thing about this, about hearing that makes it unique from vision, is that hearing information, you need to communicate very quickly across the two ears, especially to process localization of sound. And I'll get to that in more detail in later presentations. But the point is that you need to have neural connections across the two hemispheres, in a sense, across the two systems, before it actually even gets into the brain. And you can see where there's crossing over here that occurs very soon, and some processing that's done by these nuclei before it actually goes up into the brain. But right now we're just going to deal with the frequency, the sound frequency information that you're going to perceive as a sound, in the environment. And we'll neglect localization of the sound for right now and get to it later. Okay, but basically what happens is the sound information crosses over in the spinal cord, in the brainstem, crosses over to the other side and goes up into the cortex. Now, it's got the same basic configuration that you see with all sensory systems. With sensory systems, you've got a receptor that conveys to a nucleus. Usually, in this case, it's the auditory nucleus, but here you've got more than one nucleus, but to a nucleus. Then that's conveyed to the thalamus. Then that's conveyed to the cortex. Now, we saw that in vision. Vision, basically, you have receptors, you have the rods and cones, you have neurons that go to the lateral geniculate body of the thalamus, and then to the cortex. Well, here you have that basic configuration. You have receptors to nucleus, to thalamus, to cortex. Now, the processing of frequency information really doesn't happen until the cortex. Now, what this graphic shows you is that it's giving you a color coding for the frequencies, and it's using musical notation here to indicate high and low frequencies. The treble clef there, the yellow part, indicates a higher frequency, and the bass clef, which is that other symbol, indicates a lower frequency, and they've color coded that. So there's no color coding on your cortex. There's no colors there. That's, just a way of conveying that. So when the auditory nerve comes up into the cortex, you have the bass frequencies toward the outside of the brain, and then you have the higher frequencies toward the internal surface of the brain. So when the auditory nerve comes up there, it lays itself out. It communicates with neurons in the cortex according to frequency. Now, I think this idea can be captured pretty well if you think about, well, what if part of that was damaged? Well, if I damaged the outer part of the auditory cortex, I would lose low frequencies. If I damaged the more internal part, I would lose high frequencies. I would not be able to perceive high frequencies of sound. The same was true of the basal membrane. If different parts of it are damaged, you're going to get losses of different frequencies. But here again, you see this mapping, this tonotopic map of frequency based on the location of where the neurons came into the cortex. Well, this slide is depicting one of the greatest biomedical inventions in recent history, the cochlear implant. Now, the cochlear implant is designed to cure or treat deafness by supplanting a damaged basilar membrane system with an artificial one that stimulates the basilar membrane or the auditory nerve in a pattern similar to what would naturally be occurring. Now, this has had an up or down history, and it's been difficult to mimic the. You can imagine the difficulties, and just imagine the difficulties in size, having to deal with a little cochlea down there, putting a device in it that's going to mimic what actually happens with all these small microscopic neurons firing. Well, it's been basically successful. I think for people that have had, it's been very successful for people that at once had hearing and lost it, versus people that never had hearing. Because if you don't ever have hearing, your brain does not accommodate to that stimulus, so your brain doesn't develop the systems to process the sound. So even if you have a stimulus going in there from your ears, you may still have difficulty perceiving sound. Because perceiving, remember, is the construction by the brain of a perception. It's not just the input of a stimulus. Your brain goes through an active process of analyzing the sounds, processing them, and producing perceptions. So if you never had that experience, the central nervous system may be getting sound input, but it has no idea what to do with it. It hasn't had any experience at all with that stimulus. And so people don't benefit so much from cochlear implants if they never heard before. However, if people have heard before, their brains are primed for that stimulus. And if they get it back again through an implant, they can process it very well. Well, rather than go into a lot of detail about this, the course website has a great movie on hearing in general and cochlear implants. So I encourage you to watch that movie. They explain it much better in the movie than I can using PowerPoint. Okay, here I have some examples of essentially cortical level processing of a sound. They deal with basic frequency perception and some what are essentially auditory illusions that psychologists have studied through the years. There are a number of these. I won't go into detail of many of them. Here's one of the most prominent ones, which deals with the phenomenon of virtual pitch, or periodicity pitch, or virtual pitch. What's involved here is that when you hear a sound that's composed of, say here we have an example, say of A here. You have a 400 hertz tone, and then you have its harmonics. Now, I remember I've called these overtones. Harmonics is another name for overtones. Basically, you've got a 400 hertz tone here, and then at every 400 hertz above that, you've got a tone. And all these are blended together. Well, you'll hear this as a very full sort of tone, a very full sort of chord in a way. It's not technically a chord because these are just overtones, but you'll hear it as a very full sound. Well, your brain is primed to hear this. Your brain is primed to hear this kind of tone. It in a sense, it must prefer these kinds of tones because it fills in frequencies when they're missing. It wants this sort of pattern because it represents a periodicity, a very regular pattern that it commonly hears, especially in anytime anything like a pure tone is played. What happens is if you take a tone like this and you drop out frequencies, your brain will substitute the missing frequencies, and you will hear the tone like the original, even with overtones dropped out. This is very much like the Gestalt concept of good form. The full tone is good form. It's what your brain expects to hear. When frequencies are dropped out, it puts them back in. It constructs your perception so they're still there, so they're not missing. Well, at any rate, when you play these sounds, and what you do is you click on those little speakers up at the top there, you'll hear a narrator explain the example, and then he'll give you examples of this. But basically what you're hearing when he does this is sound frequencies dropping out from the full tone, and it will take you a while before you hear that they're different. A lot of the tones, the harmonics have to drop out before you hear the sound is substantially different. Until then, it sounds pretty much like the first one that you heard. And that's because your brain is filling in missing frequencies so that your perception has good form. It's harmonic. Now, in the first example that you'll play, you'll hear pretty much just what I described, where they'll play a complex tone, which consists of the bass tone and its harmonics. Then they'll take out the low tones. And your task is to figure out, okay, at what point does it seem to go up in pitch? And you'll realize it doesn't go up in pitch until, I think, the last one. The last note that's played, I thought it went up in pitch. But my brain wants to keep the tone in there. It wants to keep the pitch the same, even though I'm dropping out the low ones. Now, theoretically, if you dropped out low pitches from a complex tone, it ought to go up in pitch because you're dropping out the low ones. But because these are harmonics, your brain does not want to do that. It wants to keep things the same and wants to keep them harmonic. Now, in the next example, the second one, you're going to hear the same phenomenon, but it's going to be done a different way. Now, they use a technique called masking with noise. And what you do with masking is you play noise in the same frequency range as, say, a tone. If you do that, the tone gets blended into the noise and you can't hear it. Now, what they're going to show you is that because your brain wants to hear this melody, because it's harmonically related to the bass note, you will not let the noise confound you. You will continue to hear the melody even though the notes themselves have been masked with noise. And if you just heard those notes and the noise by themselves, you would not hear the note. You would just hear noise. But because your brain wants to hear a melody, because that's how it's been primed, it will maintain that melody through the noise, and you'll continue to hear it. In this example, we're going to see a manifestation of pitch streaming. And pitch streaming refers to the way in which pitches will integrate with each other. It's not that dissimilar from virtual pitch in the sense that we want good form. Now, as every classical musician knows, triplets, in which you have three notes together forming usually a chord or some sort of meaningful sequence in the music, are very preferred in composition. We like to hear them. We like to hear triplets put together into various forms. A lot of melodies are built around triplets being constructed using harmonics and knowledge about harmonics. Well, this example gives us a nice depiction of how triplets are formed when pitches with certain harmonics are integrated by the brain into a triplet. And here's what's going to happen in this example. When you click that speaker there, the narrator will tell you very specifically what's going to happen, but you're going to hear notes going up and down. One's going up, the other's going down. And when they reach certain ranges, they'll form triplets. You hear them, and he refers to it as galloping. They'll form a triplet. So we'll integrate them into a chord, and we'll integrate them into a little sequence of a triplet. So you hear buddy-up, 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 buddy-up. And we like that. We like that tone. Our brain does that with music. I think it's actually quite mysterious why it wants to integrate things in such an implicit way. It's actually quite mysterious how the brain constructs music to begin with. Music is not something that you need for survival value. I think it has a lot to do with how pure tones are perceived by the system. So the basilar membrane is responding to pure tones. And our nervous system is primed to process pure tones. It wants to find patterns in the pure tones. It wants good form in them. And it turns out that harmonics is a manifestation of good form. keys and all these things are representation of how we process pure tones for our own entertainment and amusement, for our own enjoyment. There's no, we're not birds where we need to have a song, that's going to help us with finding a mate or something. We have a very sophisticated implicit system for processing pure tones that we call music, that we all have at a very high level of competency, and it's not something that's related to any sort of environmental adaptation. It's quite a marvel. But anyway, this pitch streaming example gives you a good depiction of how we want to integrate tones into structures. We want them to have a melodic structure. In the triplet is probably one of the most simple, straightforward melodic structures that we use in music. And we implicitly organize sound in triplets, and this is a really good example of that.